Proteins are the molecular machines of the cells and are one of the key components controlling biological processes. There are thousands of different types within the human body, each with a specific task, location, and three-dimensional structure. DNA is the building block of the genome that directs the sequences and therefore structures of proteins. In our lab, we can manipulate DNA to create modified proteins, which we can use to study cellular processes at the molecular level. Scientists are interested in visualizing protein molecules in living cells. By doing this, they can learn about physiology and what goes wrong in diseases. Cells are a millionth of a meter in diameter, and the proteins inside 1,000 times smaller than that. So to be able to visualize living cells and single proteins, we need to use very sophisticated light microscopes. To see cells with your eyes, you can use a light microscope. Light is directed onto the sample. The scattered light from the sample is then directed using a mirror towards the eyepiece and into your eyes. The image that you then see looks like this. You can see the cell outline, but other than that, there is minimal detail. Fluorescent molecules absorb light of a certain color and then emit light of a redder color from the spectrum. For example, a molecule may absorb blue light and emit green light. In the natural world, many organisms have the ability to fluoresce. For example, some jellyfish. The fluorescent molecule encoded by jellyfish DNA is called green fluorescent protein, or GFP. Fluorescent fusion proteins are an invaluable biological tool and can be made by fusing the GFP gene to the gene encoding your protein of interest. Here we use the hormone insulin as an example. We extract the DNA encoding insulin from a pancreatic cell. Similarly, we extract the DNA that makes green fluorescent protein from the jellyfish. These two DNA sequences are then joined to create one DNA sequence that will encode our fluorescent fusion protein. If we introduce this fusion DNA into our cell, then the cell will use this to make fluorescent green insulin. This fluorescent insulin moves to the places in the cell insulin normally resides in, acting as if the GFP wasn't there. Importantly, we can now see the insulin as the protein glows green, using fluorescence microscopy to visualize particular proteins or cellular structures. We can visualize where fluorescent proteins are inside a cell. Blue light is used, exciting the GFP within the cell, which then emits green fluorescence. Blue light is also scattered from the sample and needs to be removed. For this, a special type of mirror is needed, which rejects blue light and allows the green light to pass to your eyes whilst the blue light is reflected. We use very sensitive cameras, as the light levels emitted from cells are extremely low. We can now see that the hormone insulin within these cells is stored within small vesicles, seen here moving at the cell membrane. Fluorescent imaging has provided an invaluable insight into many cellular functions within biology. However, there are still limitations. The most challenging is that proteins are a billionth of a meter, a nanometer, in size, and normal microscopy cannot see objects this small. Biologists really want to know how individual proteins behave inside the cell. This is where super-resolution imaging comes in. Super-resolution techniques emerged recently and are continually being developed. It is now possible to track tens of thousands of nanometer-sized proteins in living cells. Regular fluorescence microscopy allows for the tracking of objects hundreds of nanometers in size, like vesicles, throughout the cell, and in 3D. Super-resolution microscopy means that instead of single proteins appearing blurred, they now look sharp, revealing the exact location of single molecules a few nanometers in diameter. The advancement of microscopy has been driven by the engagement of physicists and mathematicians with biologists using fluorescent proteins derived from a range of marine organisms. This unlikely partnership has created a toolbox that allows biologists to see precisely where molecules are in cells, how they move, and where and when they interact with each other. These advances have provided us with the ability to directly visualize biological processes for the first time, opening the possibility of determining exactly what has gone wrong in diseases. New challenges include how to extract information from the huge data sets these approaches provide, and this needs the continued engagement of researchers from different disciplines. The future is bright.